How to activate T-cell. T-cells are activated when they come into contact with an antigen that is presented to them by an antigen-presenting cell APC, such as a dendritic cell or macrophage. The process of T-cell activation is complex and involves several steps. Recognition. The T-cell's T-cell receptor TCR, recognizes the antigen presented by the APC. This process is specific, meaning that each T-cell only recognizes a specific antigen. Co-stimulation. The T-cell also receives a second signal, called a co-stimulatory signal, from the APC. This signal is necessary to fully activate the T-cell. Proliferation. Once activated, the T-cell begins to divide and produce many more copies of itself, also known as clonal expansion. Differentiation. The newly formed T-cells differentiate into effector cells which carry out the function of the T-cell. For example, CD4+ T-cells differentiate into Th1, Th2 or Th17 and CD8+ T-cells differentiate into cytotoxic T-cells. An example of T-cell activation would be when a person is infected with a virus. The virus enters the body and infects cells, causing them to display viral antigens on their surface. Dendritic cells in the infected tissue, which are a type of antigen-presenting cell, engulf the viral particles and process the viral antigens. Then, the dendritic cells migrate to the lymph nodes where they present the viral antigens to T-cells in the form of peptides bound to MHC molecules. When a T-cell recognizes the viral antigen presented by the dendritic cell, the T-cell is activated and proliferates to produce a large number of effector T-cells that migrate to the site of infection to clear the virus.